ঠিক আছে।
ওকে কস্তুরী, গুড আফটারনুন। শুনতে পাচ্ছো?
হ্যাঁ স্যার, শুনতে পাচ্ছি। গুড আফটারনুন।
ওকে।
Kasturi, you may start now.
Okay. Anisha? Okay. A very good afternoon all. A warm greetings to everyone present here. It is indeed a great pleasure to have you all. On behalf of Department of Botany, Bidhan Chandra College, Asansol, I welcome you all for this one-day national webinar entitled Current Progress in Plant Biology, Implication Towards Crop Improvement. Today is a special day for our department and I am honored to announce our esteemed speakers. Firstly, we have with us Professor Narayan Chandra Mondol sir, the Departmental Head, Botany, Vishwabharati University. Our second speaker is Dr. Sanjeev Pandey, Associate Professor and Head of the Department, Botany, Banwarilal Bhalotia College, Asansol. Then Mr. Uday Shankar Rai, our next speaker, Assistant Botanist, West Bengal Agricultural Services, Zonal Drought Resistant Paddy Research Station, Hathwara, Purulia. And finally, last but not the least, Dr. Krishnendu Pramanik, UGC, Dr. D.S. Kothari, Postdoctoral Fellow, Department of Botany, Vishwabharati. Our Principal Sir, Dr. Falguni Mukhopadhyay, who is the Patron-in-Chief in this webinar. Unfortunately, he is not be able to join today's session due to some unavoidable issue, but it would not be possible to arrange such kind of events without his constant support, motivation, cooperation, and guidance. So I heartily convey my thanks to our principal sir. I extend my welcome to our coordinator, Dr. Amit Banerjee, our Bursar sir, Dr. Sujit Bera, our IQAC coordinator, Dr. Shoumen Chakraborty, our advisory group members, our guest invitees, faculties, students, research scholars, lab technicians, who are all gathered here to add some radiance to this occasion. Now, I would like to invite our IQAC coordinator, Dr. Shoumen Chakraborty, to deliver his comments. Sir, please.
Thank you, Anisha. Good afternoon to each one of you present over here. It is a moment of extreme pleasure for me to welcome all the guests and the participants who are present here to attend the webinar. On behalf of Bidhan Chandra College, Asansol, I first express my regards to the Department of Botany for arranging such a webinar. Especially both of the faculty are very young and they try to arrange such type of webinar. So it's a great thanks to them. I offer my regards to all participants. I convey my regards to our eminent speakers, Professor Narayan Chandra Mondol, Professor and HOD Department of Botany, Shiksha Bhavan, Vishwabharati, Shantiniketan, West Bengal, to Dr. Sanjeev Pandey, Associate Professor and HOD Department of Botany, BB College, Asansol, to Mr. Uday Shankar, Assistant Botanist, WBAS Research, Zonal Drought Resistance Paddy Research Station, Purulia, West Bengal, and to Krishnendu Pramanik, UGC Dr. D.S. Kothari, Postdoctoral Fellow, Department of Botany, Shiksha Bhavan, Vishwabharati, Shantiniketan, West Bengal. Thank you to all for accepting our invitation and coming over the webinar. The webinar is on current progress in plant biology, implication towards crop improvement. It is the all-time necessary and important topic for us. I think the speeches of the speakers who are quite experienced and established in this field will enlighten us, and the interactive session will also be very interesting for us. I think student participants are also very enriched by this webinar, And finally, I thank to all of you, everyone, for giving their views and opinions to make this webinar successful. Thank you.
Thank you, sir. Now, I humbly request to our audience to turn off their microphones and not to share their screens, and turn off the video to get the better quality of presentation from our speakers. Now I am trying to give a brief overview about our today's title before the speaker's valuable speech. Since 2020, we are in COVID-19 pandemic era and facing a massive socio-economic crisis and as an impact of that pandemic. The detrimental effects of environmental pollution and global climate change, along with the augmented increase in human population and decrease in arable land, have put a substantial impact on agriculture. And thus, emphasis has now been given towards ensuring long-term food supply and security for the sustainable future. With increased industrialization in residential areas, different materials, including heavy metals, are discharged into the effluent water, which pollute the agricultural lands. Besides this, during random natural calamities, the land present in the vicinity of sea facing problems of salinity stress. And there are much more problems like that. Along with these different biotic stress factors, including insects, fungi, fungal pathogens, cause various diseases in field crop and reduce yield of the plants. Among various pathogens, the fungal disease are more destructive than the disease caused by other pathogens. As the crop plants are experiencing constant threats from various abiotic and biotic stress factors, a vital route toward maintaining food security is the development of stress-resilient crops. The integration of plant-associated beneficial microorganisms may represent a promising solution to develop resistance against the various stress factors and improve the agricultural production. Furthermore, different fungi also spoil stored food, imposing new food preservation challenges, and thus various strategies are now evolving to manage and control these fungi. Now, if we come in the conventional plant breeding part, it is going on for hundreds of years and is still commonly used today for developing better quality crops with improved traits, including higher yield, abiotic stress tolerance, insecticide and pesticide resistance. And these processes mainly depend on the farmers as well as the plant breeders. But when we raise our demands with high productivity, is often found to a comparatively labor-intensive and time-consuming procedure as compared to the modern plant breeding techniques, like which add the marker-assisted selection and genetic modification to the crop improvement, which are believed to enhance the crop productivity more efficiently. So in past few decades, research is mainly focused on the developing on various genetic manipulation techniques, like their genetically modified crops, that is GM crops, or GM crops are major concerns regarding various. It is not so easily available. It is very much expensive, and it is it's slower delivery of new varieties to farmers, making it more difficult for breeders to produce varieties suited to current threats to crops. While society remains divided over the use of GM crops, new plant breeding technologies have recently emerged and alternative approaches to speed up the introduction to improve traits. These technologies include the CRISPR-Cas9 and TALEN methodologies. CRISPR-Cas9 system is a plant breeding innovation that uses a side-directed nuclease to target and modify DNA with great accuracy. Developed in 2012 by the scientists from University of California, Berkeley. CRISPR-Cas9 has received a lot of attention in recent years due to its range of application, including biological research, plant breeding, and the development of agricultural crops. In this webinar, we will acquire up-to-date information on various advancements and have been taking place for crop improvement. It is our hope that you will take advantage of this opportunity to enrich your knowledge and understanding in this area. I also look forward to open and constructive interaction after each talk. So I introduce our first speaker, Professor Narayan Chandra Mondal sir. Currently, he is serving as the departmental head of botany, Vishwabharati University, India. He did his MSc from Kolani University and then pursued his PhD from the Bose Institute, Calcutta, under the guidance of Professor P.K. Chakraborty sir. He served as an assistant professor in Darjeeling Government College from 1989 to 1993. His research interests include carbon metabolism and phosphate solubilization by common diazotrophs, survival ecology of diazotrophs under stress microbiology, water microbiology, and the studies of natural antimicrobials. He has authored more than 100 research articles in various journals of national and international repute. He is serving as an editorial member and reviewer of several international reputed journals. Dr. Narayan Chandra Mondal sir is a member of many international affiliations. He has also successfully completed his administrative responsibilities. Currently, he is serving as the Dean of the University, Vishwabharati. During his 30 years of expanded career, he has bagged many awards, including CSIR Extramural Award in the year 1986. He was a visiting scientist at Wyoming University, USA, during the year 2001. He is the incumbent president to East Zone Indian Society of Mycology and Plant Pathology since 2017. He has also supervised more than 12 PhD students till now and counting. With these few words, I would humbly request Sir to illuminate us with his talk. Today's talk is on mechanism of fungal disease control by plant growth promoting microorganisms. So please take over the screen.
Okay, Annesa, thank you very much. First, you allow me to share my slides. Please, already there?
Yes, sir.
The slides, can you see my slides?
So we don't see your slide, but you have been made co-host, so you can share it from the system, I think.
Just a minute.
So it's somewhere in the middle after chat button, sir. Share screen.
Green in color.
Yes, sir.
So
It's
देखो,
just after participants, then chat, then share screen.
सर कर्सर टच स्क्रीन ही नहीं जा
Okay, okay, I got it.
कर्सर हाँ
Screen share. Yes. Yes, can you see it now?
Yes, sir.
Visible, sir.
Okay. Okay. So, thank you very much, Annesa again. Very good afternoon, everybody. I respected Principal Sir, although he is absent, Professor Falguni Mukhopadhyay, Chairman IQC, Dr. Somen Chakraborty, the coordinator of this program, Dr. Amit Banerjee, and my dear Annesa Bandyopadhyay and Kasturi Chattopadhyay. So, although they are very young, but I am overwhelmed by their enthusiasm. See their enthusiasm, and they are from the very beginning. They have taken this initiative, and now they are in the driver's seat to run this program with so efficiency, and it has already been started. So I am very thankful to them, and I am very much thankful to the organizers for inviting me, for allowing me to share some of my experiences on this line of talk. And I also address my very academically dynamic younger brother and friend, Dr. Sonji Pandey in today's program. And
Good afternoon, sir.
yes,
Sanjeev Puri.
very happy to see you here in this program.
Me too, sir.
Uh, uh, Dr. Uday Shankar Rai and Dr. Krishnendu Pramanik is my student now. So all they will deliver to enlighten today's program. I do not know how much I can enlighten you, but frankly speaking, my presentation is not so organized because I am technically very weak. I do not know how to prepare very good slides. Anyway, so whatever my experiences on this field, I would like to share it with you. So based on theoretical points and based on some experimental parts. So I'll speak on mechanism of fungal disease control by plant growth promoting microorganisms. So there are many questions on it. So one by one, I will come to these points. Slides are not moving. Can I go to the next slide?
স্যার ফুল স্ক্রিন করে নিন।
Okay. So we know all that fungi are at the top of the list among plant pathogens. As we are talking about the crop plants and plants, I'm coming to plants only right now. So fungi, we know that fungi are at the top of the list among plant pathogens. Whatever may be the thing, the biotic agents who are the sufferers of or who can cause sufferings to the plants, fungi are at the top of the list. If you take any survey or if you do any test, so you can get always this result. How? Because they have the ability to directly break open the cuticular and epidermal layer by their physical infection peg. It can puncture open by physical force with the help of ATP driver physical force, and by their enzymatic property, which can degrade the cellulose, this pectin, this lignin, all these layers. No other plant pathogens like bacteria, viruses, they can puncture, they can break open this layer. So fungi have this capability. And because of that, fungi are at the top of the list among plant pathogens. And another thing, although I do not believe all the times, and in most of the plants, they have a matching body temperature. Like for human beings, they have 37 degrees centigrade temperature, and most of the human pathogens, they prefer to grow at 37 degrees centigrade, and most of the plant pathogens, they prefer to grow at 28 degrees centigrade, and most of the pathogens, and in most of the common mesophilic plants, their body temperature is around 28 degrees centigrade. And if we take a statistics or go for a survey, then we can find that fungi are at the top of the list among all the disease. If you analyze all the disease, all the causative agents, then you will find only fungi and fungi. And next, not only crop plants, fungi can cause immense damage to the timber plants, woodlots. And if in this particularly this time or during winter time, during the Mukulasar Samoy, you can see that many mango plants get totally wilted, wholly. So there are so many trees which are totally damaged by these fungal pathogens, and these are mostly by these Basidiomycetes polyporous members fungi, and they cause white rot and brown rot diseases. And not only that, they are responsible for post-harvest spoilage of various crops, whether it's a grain crop, it is a pulse crop, oilseed crops, or whatever it may be. So fungi are responsible for post-harvest, even for fruits, vegetables. They do post-harvest damage and are responsible for major crop loss. And they produce potentially dangerous mycotoxins in the food grains and cause health concerns of ours. So it is mandatory to control them. So fungal control is essential. So what are fungi? Basically, morphologically, we can see fungi in nature in three forms. Some are yeast, very microscopic, which can reproduce by budding process. Then they are elaborative form with mycelial form, or which are known as molds, and the filamentous structures. And these filamentous structures, when further developed or go for evolutionary organized, they are more organized, they form these mushroom structures. And whatever it may be, all true fungi, they have some unique properties among the eukaryotes. And these properties are not shared by any other eukaryotes, plants or animals. These properties are absolutely confined to them only, to fungi only. Fungi have hyphal tip growth. They grow by hyphal tip, apical tip growth. They have chitin and beta glucans in their cell. No other organisms among eukaryotes, like animals or plants. Plants have chitin, but in their exoskeleton, not in their cell wall. Animals, they do not have any cell wall. Okay, so animals, they have exoskeleton chitin, but fungi have only chitin. And presence of ergosterol, it is a special kind of membrane lipid present only in fungi, and in contrast to cholesterol in human beings, cytosterol in plants, and hopanoid, it is a sterol-like substance in prokaryotes. They have heterotrophic absorptive mode of nutrition. You know that among the in Whitaker's classifications, only the multicellular heterotrophs are Animalia and fungi. And Animalia, they have they take food inside their body or coelomic cavity. Their digestion takes place inside their body, but fungi, they do not take food inside their body. They digest. The food is still remain outside. So fungi have stomach outside their body. They digest food is when it is present outside their body. So it is a property of fungi. They have degenerative mode of sexual reproduction. Fungi, they have nuclear membrane, the persistive nuclear membrane during cell division, in contrast to plants and animals. And fungi have microtubules, which are insensitive to colchicine, which is contrasting characters to plants and animals, but which are sensitive to graciofulvin and benzimidazoles. And fungi have lysine biosynthesis by alpha amino adipic acid pathway. And fungi can code for tryptophan by a nonsense codon of human beings or animals or plants. It is UGA, it codes for tryptophan, particularly the mitochondrial codon usage. We can get tryptophan by UGA, it is a nonsense codon. So these are the properties of fungi. And we know that according to many scientists, particularly the German scientists who point this chemotherapy, where the selective toxicity or permeability is there. So if fungi have these properties, and if we do not have these properties, we human beings and we plants, they do not have these such properties, then we can design a drug or a molecule which can hit only at these sites. And literally on and for all human medicines, to control fungal disease, we can target at ergosterol metabolic biosynthetic pathway. And for plant disease control, mostly we target at chitin and beta glucan biosynthesis or at these sites. And scientists are trying to address some other parts of the these unique properties of fungi. So how can and what are the materials till that used in fungal disease control? And these are the different sites here in cell membrane. Okay, I do not particularly. This is the common antifungal classes. If we for plants, for animal, what kind of the pathogens? Fungal pathogens. We can kill these fungal pathogens by using such compounds. Polyenes. They bind with ergosterol. Ergosterol is a very essential membrane lipid of fungi. So polyene antibody like amphotericin B, which is nowadays used for controlling black fungi of post-COVID problem. So polyene amphotericin B, it is used. So it binds with the ergosterol, it removes the ergosterol, it makes pores in the fungal cell membrane, and the internal content of the cell membrane, fungal cell, the electrolytes, the water, everything come out, and the fungi are being killed. So polyene antibiotics. Then azoles, they are fungistatic. These polyenes are cidal. These azoles are static, and so many azoles are there, like fluconazole, itraconazole, ketoconazole, and voriconazole, posaconazole. All these azoles targeted at ergosterol biosynthesis and membrane lipid biosynthesis. So if we apply azole, we can control, we can hamper fungal cell membrane biosynthesis, proper functioning of the cell membrane. Then echinocandins, these inhibit the glucans. In case of fungal cell membrane, beta glucans, cell wall, beta glucans are very essential. And echinocandins inhibit glucan by inhibiting glucan synthase enzyme. Then allylamines, it is another ergosterol biosynthetic enzyme inhibitor. Then nicomycin Z, nicomycin can inhibit casein biosynthesis. All these are antibiotics. Then sodarins, they inhibit some fungal wall proteins. And then N-myristoyl transferase inhibitors. These are very important proteins. In this myristoyl compound that binds with the glycine molecule, a terminal end of the glycine molecule, and responsible for signal transduction in fungi, particularly in pathogens, and these inhibitors can stop these functions. And similarly, these phingolipid synthesis inhibitors are also important. So these are in general the antifungal antibiotics nowadays working with full targeted, and particularly the scientists are doing, and so that they do not cause any harm to human beings or plants. But unfortunately, this polyamphotericin B, the first one, it has tremendous this polyene, particularly these amphotericin B, it has tremendous nephrotoxicity, renal toxicity, and scientists are trying to overcome this problem, but still we do not have a better answer to solve this amphotericin B toxicity. So then we depend on these ergosterol metabolic inhibitors, and this is ergosterol biosynthetic pathway. It starts from acetyl coenzyme to ergosterol. So many intermediates are there, and so many ergosterol. Most of these are azole compounds. These ergosterol biosynthesis inhibitors, allylamines, terbinafine, all different kinds of molecules are there. They target at different sites of ergosterol biosynthesis, and we can stop ergosterol biosynthesis, and we can control fungal growth. And these are different types of azoles, different types of how can this azole inhibit fungal important metabolites, metabolic processes, and fluconazole, voriconazole, all these. I'm not going in detail because most of these are used for human treatment, and my concern is for plant disease. So just to give an idea, just overview, I am giving these names. This squalene epoxidase inhibitor is a very important compound at early stage of our ergosterol biosynthesis. This squalene epoxide molecule is formed, and if we can stop its biosynthesis, we can control fungal membrane development. So these are echinocandin. They stop fungal cell wall biosynthesis, particularly this caspofungin like compound. So caspofungin and echinocandin, all these are used for human treatment, and these are nicomycin Z, particularly work in chitin biosynthesis process. This nicomycin Z, it inhibits myosin chitin synthesis, and it stops this chitin assembly in a fungal cell wall. So nicomycin Z is a beautiful antibiotic, and its production cost is very high. So it is not used for plant disease control, but azole drugs are very cheap. So azole drugs are, lots of azole drugs are used in fungal disease control. And as we apply lots of azole drugs in the field, and probably, and these fungi which are otherwise normally used to stay in the field condition, either as saprophytes or in the plant pathogens, they become resistant to azole drugs. And when they come to an immunocompromised person and they cause infection in human beings like black fungi, mucor, rhizopas, all these with azole resistance properties. And then these azole drugs like voriconazole or posiconazole, which are common systemic fungicides in human beings, are not at all useful in controlling those. So we are bound to apply this amphotericin B. It is a cidal antibiotic, keeping in mind that these may cause renal toxicity. Anyway, so this will be another thing. It can arrest spindle cell wall, spindle microtubule breakage. It is responsible breakage of microtubule and cause polyploid formation and cell division. It disturbs cell division. And anyway, so now we should come to plant disease and plant pathogens. So I have given a glance of antibiotics which are responsible for killing fungi and mostly are used in plant disease control, human disease control or mycosis control. And our concern is plant disease. Today's topic is on plant disease. So And I have already mentioned that for most of the plant disease control, fungal control of plant disease is responsible for plant disease control, targeted at cell wall biosynthesis. And what are the components of cell wall? Cell wall is consist of chitin. It is a polymer of acetylated amino acid, N-acetyl glucosamine, then glucans, beta-1,3 glucans or beta-1,6 glucan, and some proteins. And these are present in this form. These are the nanoproteins or different kinds of proteins are there in the cell wall. And these are beta-glucans or beta-1,6 glucan or beta-1,3 glucans or glucomanons are different forms. They are mixed in different forms. Then chitins are there, and at below cell wall, cell membrane is there. So cell wall is very important. And in case of plant pathogens, fungal pathogens, and mostly the crop scientists or the mycologists or the plant pathologists, they targeted either on this beta-glucan or at chitin or at these proteins. So how, if we can degrade these structures of cell wall structures, and we can stop the biosynthesis of ergosterol, then we can easily manage fungus, fungal growth or pathogenic growth. And these are the chitins. When it is deacetylated, N-acetyl glucosamine, when it is deacetylated by acid treatment or by natural process, then it becomes chitosan. And chitosans are mostly present in zygomycetes type of fungi. And we know that zygomycetes, these are like Rhizopus, Micor, they are zygomycetes. Cunninghamella, they are zygomycetes. And these cellulose are present in oomycetes in Staminophylla group. And the beta glucans which are present, the glucan which are present either in the beta 1, this is 1 position, and this 1,3 linkage, or this 1 position and 6 position, this is 1,6 linkage. And therefore either the chitin are degraded by 3 different types of chitinases enzymes, and one chitinase, 2 chitinases, they are exochitinases, and 1 chitinase, they are endochitinase. And then before coming to this slide, just I have something to say. I do not know who are the participants in this today's program. I think many honor students are there. Many students are there who are not from plant pathology and not microbiology. So they have a initial, even I have also some knowledge that we can control plant pathogens by organic and inorganic fungicides, like in the inorganic, these Bodo mixture, copper fungicide, mercurial, sulfur compounds, all these things. And organic, these dithiocarbamates are there, and these organophosphates are there, and these compounds are there. The heterocyclic nitrogenous compounds are there. The different, this type of compounds are there, responsible for different, they have different mechanism, and all these are not very bad also. And the systemic fungicides are there, benzometazole, the oxanthins are there, different molecules are there. And they are these chemical fungicides or chemical components which can kill fungi. And we have no problem in these. But when we apply these compounds have enormous toxicity in human system, in mammalian system. They cause lots of damage. Many are xenobiotic compounds, they are carcinogenic compounds, and they cause a lots of problems in human system or animal system. So we must avoid it. And I do not have any scope to discuss all the abnormalities they are creating in human body. So we have to be very careful for this. So we scientists are targeting some alternate mechanism, and of the different types of alternate mechanism, some hyperparasites are there. And we know trichoderma is a very good hyperparasite. What is the meaning of hyperparasite? A microorganism which can parasitize the pathogen, which can parasitize the pathogen of a crop plant. তাহলে একটা crop planter pathogener উপর যেটা grow করবে বা তাকে kill করবে, that is known as hyperparasite. So basically hyperparasite can kill a pathogen of plant. So planter pathogen ke hyperparasite kill korbe by disintegrating its cell wall structures or chitinase and beta glucanase or different types of glucanase productions. Or they can compete with the pathogens by their nutrition snatching process through sideropores. And they can produce antimicrobial compounds. So basically, these hyperparasites produce antimicrobial compounds. They produce enzymes for degradation of their cell wall materials, or they compete with the pathogen by for very essential macroelements like iron through sideropores. So these are basically the essential criteria of a hyperparasite. I will be speaking something more, that is plant growth promoting microorganisms. We can add some value in hyperparasite. Definitely the PGPR or plant growth promoting organisms are hyperparasites. But what are hyperparasite? What are plant growth promoting microorganisms? Sorry. These plant growth promoting microorganisms, they are microbes functionally equivalent to the probiotics, probiotics of human. So lactic acid bacteria which grow in our gut system, so they are called probiotic. They help in human body, human health, and thus considered as plant. They are considered, typographically I mistaken to put this. So they are considered as plant probiotics, and they are endophytes, rhizobacteria, hylosporic microbes, and different types of PGP organisms are considered as different types of plant growth promoting microorganisms. So I'll be speaking on these. They can contribute directly to plant growth by producing phytohormones, and this is not a must criteria of the hyperparasites. So PGP, they produce IAA, gibberellic acid, and ACC deaminase, and they can mobilize nutrition to the plants. So they can fix nitrogen, they can solubilize phosphate, they can solubilize potash. So NPK is the essential requirement of any crop plant. So if you go to any farmers, they are applying NPK. So nitrogen, phosphate, and potash, these are very important. And these PGP, they can give these things from unavailable source to make them available. So biomondolage nitrogen, so unavailable nitrogen, so N triple bond N, which cannot be utilized directly by the plants. So it must have to be fixed into this compound form ammonia. So it is done with the help of nitrogenase enzyme. So PGP organisms contain these things. They have this solubilizing mechanism. They can convert insoluble phosphate into soluble phosphate. They have convert insoluble potash into. So these are another criteria. They can chelate iron through sideropores. And we know that all aerobic organisms need iron, because without iron, their ATP, their electron transport chain cannot operate. The cytochromes cannot operate. So they cannot help in transport of the electrons, and ATP will not be generated. And sideropore will help in acquiring this iron. And these PGP organisms, they help in giving sideropores iron through their sideropores to the crop plants. They can kill the pathogens. This is definitely this is essential criteria of hyperparasites. And these PGP organisms also produce these antimicrobial compounds by by their antimicrobial metabolites, and by their antimicrobial enzymes, and by their induced ISR, the induced systemic resistance. They can induce the immunity to the crop plants. And this is diagrammatic structure. These this PGP organism, they can also combat abiotic stress by various mechanisms. I do not have much time to discuss all those things. So now I am giving 3 different types of examples. One is a PGPR, plant growth promoting rhizobacteria, isolated from this Lycopodium root system, and a number of organisms are isolated. So bacteria, they are Bartholderia basically. Then one endophyte isolated from Serraritinum, and they are identified. This endophyte is identified as Bacillus siamensis, and their morphology are this. We can see this is a gram positive. This is violet color gram positive. Electron microscope, they are rod bacillus, and they have these properties. And all microbiology students know that these have catalase test, Hooks-Proskauer test. There's a methylene test, indole test, different types of tests. Then after that, we can go for their molecular identification through 16S for prokaryotes, and we can isolate their 16S gene, and we can PCR, we can amplify it through PCR, and we can sequence it, and with the sequence, we can compare it with the library and CBA database, and we can get the identification of and the Lycopodium rhizosphere bacteria, they are Bartholderia unami, they are Bartholderia cepacia, and they are Bartholderia tropica. And these these are endophyte bacteria that is known as Bacillus siamensis after this 16S identification. So they are also, and I have already mentioned that their mast properties are hormone production. So we have checked that they are pretty enough, they are pretty efficient in producing indole acetic acid, even in without tryptophan. Tryptophan is the precursor element, and even with tryptophan and without tryptophan, they can produce indole acetic acid. And these are the indole acetic acid test, different types of test. These are bioassay, and you can see that indole acetic acid, if you apply indole acetic acid, the excessive root branching will be there. So you can see that root branching is here. So indole acetic acids are produced by these organisms. So indole acetic acid is produced, is confirmed by HPLC test, and all these experiments are done by my students under my direct supervision. So they have done all these experiments. These are my direct work from my laboratory, and SEC deaminase got this. Please knock me if I will ask 5 minutes more time, because I have some more slide. So ACC deaminase is very important enzyme. It is known as ethylene management enzyme. Normally, we know that ethylene molecule is produced from ACC, 1-aminocyclopropane, 1-carboxylic acid. Ethylene is produced under stress condition or under various conditions. Ethylene is produced, and once ethylene is produced, it will propel, or it is compulsorily put the plant into their senescence phase, and they will lay a lot of sufferings. And if there is SEC deaminase, then they will convert this precursor molecule into alpha ketobutyrate and ammonia, and ethylene will not be produced. So it is a very important marker molecule for a good probiotic or good PGP organism. So, the organisms, if you want to, if anyone want to isolate any good PGP, then that organism must contain this SEC deaminase property. So after these enzymatic properties, and then we have checked that normally we know that nitrogen fixation can be assessed by acetylene reduction test. But in our laboratory this time, I do not have the slides. Earlier I did many ARA assay of many PGP organisms, but right now here we can test that in nitrogen-free box medium, when the organisms, it is the highest purity quality. When the organisms can grow in nitrogen-free box medium, we can assume that organisms can grow in nitrogen-free medium, and it is a phenotype of their nitrogen fixing property. This is indirect property. Direct property is acetylene reduction assay. Then their phosphates, nutrient solubilization. All these white, fog color, this white color thing is insoluble phosphate, and the organism is given at the center, and you can see this transparent zone is the indication of phosphate solubilization, and it is another test of phosphate solubilization study. And here we have this Bartholderia cepacia, which is isolated from these Lycopodium roots, and they have isolated 580, 570, 485 microgram per ml phosphate they produced, and we know if it is more than 300 microgram soluble phosphate is there, it is pretty potential phosphate solubilizer. So these are pretty phosphate potential solubilizer, and the commercial strains, they have produced this amount, and our strains are much better than the commercial strains. And the organisms are also very efficient in forming biofilm on the phosphate granules, and these are electron microscopic photographs. And they produce these siderophores. This orange color in a chromazural agar medium. If they form this orange color zone, it is a clear indication of siderophore formation, and it is a tetrazolium test, and it is hydroxamate type of, there are different types of cytoreports. And if I find any time, or if anybody is interested, he can directly interact with me through my email. I can give answer, any kind of answer. I am ready to give, and I'll be happy to answer all the questions. So if you have any particular question on any aspect, I'll be very happy to answer you. And so there are many 2-3 different types of this hydroxamate, then catecholate, then phenolate, different types of cytoreports are there. And here this cytoreport is obtained from Bacillus siamensis, the Aerobic, and then they have this CNE6 Bacillus siamensis, and it has tremendous antifungal property, so antipathogenic property, and again, these are the Lycopodium rhizosphere PGP, and they have antipathogenic property, T pathogen Pestalosia 3. And if we put their extract, cell-free extract in a medium and apply fungi, and if we do not apply any PGP extract, the fungi will grow happily. And if we increase the concentration of PGP extract, cell-free extract, then it can stop fungal growth. And you see, it is how fungi grow radially, and with the increase of the cell-free supernatant, it can stop fungal growth. And if we apply the supernatant and take photograph, and it is the plant growth promoting organism isolated from Lycopodium, and it is applied against Pestalosia, the T pathogen, and just see how the mycelia get damaged by pore formation. The mycelia is totally degraded. This mycelia totally almost chewed up. Mycelia is totally degraded. And another example, so I have so many examples, and all the papers we have published in either in Biological Control Elsevier or Microbiological Research Elsevier, or in different good journals, scientific reports, all different journals, Chemosphere, all different journals we have published. And here it is a this CNA6, it is isolated from Scissera retinum, and Scissera retinum has a very deadly pathogen which is known as black root disease, and the pathogen is known as Fusarium. And this you can see the Fusarium, they produce this macroconidia and microconidia. These are macroconidia and these are microconidia, and the Fusarium pathogen is identified also. It is Fusarium solani, and they cause disease, this black root disease in a aseptic condition. We have, this is the normal plant, and here pathogen is added, and here the normal plant, here pathogen is the black root disease. So all Scissera plants get diseased if the pathogen is there, and the pathogen can easily be controlled by this Bacillus siamensis. Here it is a dual culture overlay assay method, and each and every experiment I do not have much time. So if anybody have any questions, so I will answer all the questions. And here again we have take scanning photograph, and the mycelia get totally disintegrated. This is the normal mycelia. This is Bacillus siamensis. This PGP extract is added here, and the mycelia gets chewed. And the pathogen on the host surface, they form by this biofilm. And if we add this PGP extract, then the biofilm of the pathogen is not formed. Just if we increase the extract of the PGP, and then their compound, their biofilm will not be produced. So, and again, the organism Bacillus siamensis, they can produce many antifungal compounds, antifungal metabolites. And here you can see this is surfactant, this is ethylene, this is fungicin. These are very potent antifungal compounds, antifungal secondary metabolites. They are produced this pathogen. And these are by LCMS data. We have isolated, extracted, purified, and tested. This is only the purified final result. And before this documentation of this result, we have so many earlier results. I am not showing all those things. And the genes are present in the Bacillus siamensis, the PGP organism. We have tested all that these genes are also present here. And using proper primer and of this surfactant, ethylene, fungicin, we have checked that these genes are present in the host, in the PGP organism. And then we have applied. So these are these are plant experiment, and we have applied these PGP organism, the plant growth promoting organism, and we could see that they can very easily control these diseases. And many people may raise question that how can you sure that this bacillus, this organism is tracking into the plant? Tara planter moddhe dhukche, tumi ki kore sure hobe? Then we went for anyway, and they also induced some. I'm coming to that point later. And they induced some defense genes, and we have checked that. And these are the different types of defense genes: chitin, chitinase, glucanase. Then peroxidase. Different types of defense genes are expressed in presence of this chitinase. These different types of, these are defense genes, and these defense genes are induced in due to induction of these PGP organisms. And people may ask that how can you be sure that your organisms are tracking into the plants? I will be showing that later. And I have just I know that my time is already over. But we have attempted to take chitinase, as chitin is very important. So chitinase from different sources, and we have targeted another source, that is chitin consuming animals, their gut bacteria. So tiktiki, this is a very important house gecko, and this is another. They live on the fish, and these are the we have exoskeleton of the animals, so live on chitins. And from their gut, we isolated different types of chitinase and glucanase and protease. These different types of enzymes we have isolated. And these organisms have identified Aeromonas veroni and Bacillus subtilis NSS. These organisms are isolated from this tiktiki and this fish fly. So these organisms are, we have sequenced their whole genome. And from their genome, we got an idea that what different kind of genes are there, and these are different types of genes are there, and from this we can different types of I would be interested to answer after this seminar. I will be available. So on this aspect, I will be ready to answer. So from whole genome, it is used by an NGS platform, and whole genome can be sequenced. And from which sequences we can get different types of metabolic cascades, metabolic genes, like phosphate solubilization genes, like carbohydrate metabolic genes, like DNA replication genes, like different types of, like lipid metabolism gene, translation gene, transcription gene, drug resistance gene, all different kinds of genes. What are the genes present in the total genome of the organism? We can get an idea of that. And from that, we have targeted particularly our interest, the antifungal enzymes, and of the different antifungal enzymes, these chitinase, this protease, and these beta glucanase enzymes we have initially targeted, and then we have targeted the PGP traits, and then we have targeted the metal properties, and these are big stories, and I'll not go in detail for that. And these are different types of chitinase enzymes, all their different properties: amino acid length, molecular weight, isoelectric point, and these different enzymatic properties, and all these are validated with different types of bioinformatics tools, and later on by benchmark by wet lab experiments, and we have isolated. We have this is a model of chitinase, a particular chitinase, and we have got 7 different types of chitinase enzyme, different types of protease enzymes, different types of beta glucanase enzymes. We have compared our chitinase enzymes with the existing chitinase enzymes, and we have found this Aeromonas CMF, Aeromonas veroni enzyme with other organisms, and they matches with the other organisms. And then we have a different family chitinase. Normally these chitinase enzymes present in plant chitinase, and we got this chitinase in NSS and in CMF. So our organism, although it is a bacteria, in gut bacteria, but still it possesses the plant family chitinase. Normally, it is a PR protein which is induced in plant. That chitinase is available in plant. It is a novel finding of ours, and these chitinases are also found in these enzymes. And we got a chitin metabolic pathway from and from based on KEGG database. There are so many software, many bioinformatic tools and techniques using these and combining with your wet lab experiments and with this software database and whole genome. If you have a whole genome database in your hand, you can design so many experiments. And so we got, and later on we got one beta 131 glucanase enzyme, and we made an recombinant strain using that, and I have very less time. And anyway, so we made one overproducing strain. For all these contribute a lot in degrading the fungal cell wall. So basically, whatever may be the source, it may be a rhizobacteria, it may be endophyte, it may be a gut bacteria. So we can get chitinase or glucanase or protease. So they can be utilized for combating the deadly pathogens of plants. So and they have a lot of cassettes for this is phosphate solubilizing cassettes, and these are the siderophore cassettes. These are different types of other enzymes. These are IA producing cassettes present in the whole genome, and we can do many experiments, and we can confirm all these through our wet lab studies, and we can as we know that the genes are present in the organisms, and so we can confirm these phenotypes through our wet lab experiments. And again, we confirm that we have an enterochelin type of siderophore in that organism, and later on we have confirmed that these organisms can track into the plants through these fluorescence studies, and using GFP tagging, green fluorescence protein tagging. We tag these proteins into the PGP organism, and we have through these experiments we tagged it, and we followed that inside the vascular bundle, inside the tissues of a particular plant, these organisms are tracked into the ideal plant under aseptic condition or under the control condition, so that no other pathogens are there. And the other organisms, if it is present at all in the environment, they are not tagged with GFP, so their existence will not be monitored. Only the GFP tagged organism can be monitored. So as we tagged our PGP organism, and as these are tagged through confocal electron microscopic study, confocal microscopic study, and so we confirm that our organisms are responsible for controlling this. So anyway, so all those things. I have another approach, but I will not discuss. Some human probiotic organism can also help these things in plant disease control, and they have enormous potential in killing the plant pathogens. We have tested in our laboratory that they have enormous property potential to kill pathogens of plants, and these plant pathogens get degraded, their mycelia get degraded, and you may have a question: why lactic acid bacteria, which are human probiotic organisms, can help plant? And because one answer is there, because plants produce a lot of prebiotic compounds, and these prebiotic compounds invite the human probiotic organisms to grow onto their surface. So human lactic acid bacteria or probiotic bacteria in many occasions grow on the plant surface, healthy plant surface. They create many compounds which can kill the plant pathogens, particularly the fungal pathogens. So with this, I would like to conclude my talk here. And the basic objective, basic thing is, basic message is that for plant disease control, fungal plant disease control, we must target it. Right now, we have the mechanisms to break their cell wall structures mainly, and we have enough potential if we exploit the plant growth promoting organisms. And plant growth promoting organisms have enormous potentials, and I have shown it taking examples from various domains. And with this, I hope, although it is a big elaborative topic, and I am very sorry to put a large topic into a small time, and I beg apology for this. And with this, I handing it over to the organizers. Thank you for your attention.
Okay, so thank you. Thank you for sharing your experience with us. We are running short, so we have some questions in our chat box. May I take one question here, sir?
Okay, you tell.
One question from
Any question?
Orno Broy. He says, few days before Aspergillosis fungal infection cases found in India. What is the Aspergillosis fungal infection?
This is very common when human beings' immunity status get lowered down. But there are so many ways for lowering the immunity. Recently, because of this corona, in corona treatment, this treatment with steroid is very essential, and to lower down the cytokine storm. So steroid treatment is very essential, and if we apply steroids, so our immunity will be lowered down. And with immunocompromised persons, so there are many saprophytes, many fungal opportunistic pathogens. They come not only Aspergillus, so Candida will be there, Rhizopus will be there, Mucor will be there. They come into human body, and they will grow in human body, different human tissue, and they use human tissue as their growing substrate, and they cause disease. They grow happily on the human tissue, and they cause disease, and they cause, if it is Aspergillus, it will be Aspergillosis. If it is Mucor, it will be mucormycosis. If it is Candida, it will be candidiasis.
Okay, sir, thank you. To the audience, I will say, for your further inquiry, you can put your questions via mail. The mail ID of the speakers are already have been put into the chat box.
So you don't worry, if today is not possible, you put your question, you write into my email. So I will answer all the questions. I will answer everybody's questions, no problem. I'll answer every question. So put questions in my email and no problem. Okay.
Okay, sir. Okay, sir. Thank you.
I
Thank you
am
once
very
again.
sorry, Sanjeev bhai. I have
No, sir, no,
I have
sir.
snatched your time.
No, sir.
No
Thank you.
problem,
Thank
sir.
you
It
once
was
again.
really informative for me also. Learned so many things. I will apply those in my lab if possible. Thank you very much.
Thank you, sir. We are moving to our next lecture, and I'm handing over the responsibility to introduce our next speaker to our convener, Kosturi Chatterjee. Kosturi, over to you.
Thank you, sir. Thank you, Anisha. So now I would like to invite our next speaker, Dr. Sanjeev Pandey, HOD and Associate Professor, Department of Botany, BB College, Asansol. If we look at his career, he was a gold medalist during his master's, and he completed his PhD from the University of Bardhaman under the guidance of Professor Tushar Kanti Maiti. He has worked for a short period of time at IICB Kolkata. He possesses immense experience of almost 20 years in the field of teaching and 24 years in research, and still counting. During this period, he published many research articles in different journals of national and international repute. He has also authored and edited a plant biology textbook, mainly focused and conceptualized for the undergraduate and postgraduate students. He has been the PI for one UGC minor research project and one WBDST major research project. His current research interest is mainly focused on PGPR and soil microbiology. Today, he will deliver a talk on the topic CRISPR-Cas, a magic tool for crop improvement. Now I request him to enrich us with his talk. Over to you, sir.
Thank you, Kasturi. Can you hear me?
Yes, sir.
The voice is audible?
Yes, sir. Voice, video, both clear.
Everything is clear. Okay, let me share my screen now. Screen is visible? No.
Yes.
So Kosturi, you caught my sharing.
Okay, no, my screen is visible
No,
now.
sir. No problem.
Okay.
You can see my screen.
Yes, sir.
Yes, it
Yes,
is
sir.
very audible. Very visible. No problem.
Okay, sir. So let me go to the first slide. Okay. So first of all, a very good afternoon to all of you. Respected Sir Dr. Narayan, Professor Narayan Chandramundal, sir, my senior teacher, my Guruji like, and senior brother like. A very good afternoon to the IQSC coordinator, to the coordinator of this webinar, and the very young organizers, Kasturi Chatterjee and Annesa Bandyopadhyay. They have taken I think it's a difficult job they have chosen in this young career. I haven't been able to do such kind of seminar, even in 20 years of my experience. I always think it is very difficult to organize such a type of national webinar. So I'm going to start my lecture from this title, CRISPR-Cas, a magic tool for crop improvement. This is a very burning topic, as you know, and it won 2020 Nobel Prize in Chemistry to these two young women scientists, Emmanuelle Charpentier and Jennifer Doudna. One is from France, another is from US. So I will divide my lecture into three parts. The first part will deal about the basic idea about DNA and gene expression. Then we have to delve a little bit about the historical development of CRISPR-Cas to understand it better, that how it developed into a genetic gene editing tool. Then I will go for the application part. I will try to brief it as far as possible, since time is limiting. So the first part, as you know, that the cells are made up of our bodies, every organism's bodies are made of cells, and cells they may contain nucleus or they may not contain. If they're prokaryotic, they will not be containing nuclei. But each of these cells have the chromosomes, and the chromosomes contain the DNA contains the gene. This part is, I'm dealing this basic part just for those people who are not from biology background. The genes are the small segments of DNA. They are not the entire DNA that is present in the chromosome. They're only small portions of the genome. You can imagine that human genome has only 3% of total genome is gene, less than 3% even. So these areas of DNA, they have a meaning. They actually are responsible for the synthesis of proteins, and they determine our character. So you can imagine that genes are the hereditary units in simple words. So when suppose this picture, the genes are made up of the four letters, the nitrogenous bases, A, T, G, C, adenine, guanine, cytosine, and thymidine, and different type of sequences might be present. So this is double-stranded DNA, the black color, and you know that T comes against A, and C comes against G. A versus T, G versus C. This is the complementarity. This way, the double-stranded DNA is hybridized or present in a helical manner in our cells. When RNA is synthesized from DNA, it uses one of the strand as template, and the other strand is the R template. If you see that the template, the sequence of the RNA is same as that of the sequence of the R template strand, only the T will be replaced by U in case of RNA. Otherwise, everything will be same. A, C, G, C, C. In place of T, it will be U, G, G, A, C, C, like this. So the RNA, in the next slide, we will understand that why I'm telling this. The information concealed in the DNA in the form of genes are transferred finally to the proteins via RNA. And these proteins are our final expression via which we get our characters, different kinds of characters that you can see of an organism is because of the proteins. The sequence of amino acids in the proteins are determined by the sequence of ribonucleotides in the RNA and deoxyribonucleotides in the DNA. So for each of the amino acids in the protein, there are 3 nucleotides designated for that, for each of the amino acids. So if a protein is made up of, say, 50 amino acids, there will be 150 nucleotides for that in the RNA, and there will be 150 deoxyribonucleotides in the DNA. So this is the Codon Dictionary. Scientists have deciphered that which particular genetic code. These are called genetic code. These triplet sets of nucleotides, they are called genetic code because each of them code for a particular amino acid of a protein. So this codon dictionary helps us to understand which particular codon is responsible for which particular amino acids. This concept, this flow of information occurs from DNA to RNA to protein. This is called central dogma. These things are required for our basic understanding of this critical topic, CRISPR-Cas9. So let me just define first what you're going to discuss in this, what we're going to explore in this talk. CRISPR-Cas9, what is it? In its present form, CRISPR-Cas9 is basically an RNA-guided endonuclease, an enzyme that can cleave nucleic acids from inside. Endo means from within the DNA. And it has got the capacity to identify its target DNA and introduce very, very specific, very precise cuts on the desired position in the DNA. So this is the RNA that we are talking about, the single guide RNA. So you need a guide RNA, and this is a protein that is called Cas protein, and it is going to find its target DNA. This is the double-stranded DNA where it will introduce a double-strand break, and you are going to a magical result after that. So how did we reach to this CRISPR-Cas9 magic tool? Actually, this CRISPR element is present in the bacterial system. It is in the bacterial chromosome. This CRISPR, it comes from this bacterial, the full form of that is, it's a jugular term, the full form is clustered, regularly interspaced, short palindromic repeats. So it's a big, the abbreviation is CRISPR. So if you try to analyze each of the word, you get some meaning. Short, you understand what is short and repeat. This is very easy to understand. These are repeated sequences of DNA. Palindromic means they read, in forward and backward direction. If you read a particular sequence, and if it is same in forward and backward direction, we call it a palindrome. Suppose from 5' 3', you take the sequences T, T, C, 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 N, N, G, 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 A, A. The other strand will also have 5' T, 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 C4, N2, G4, A2, 3' like that. So this is a palindromic sequence, and these kind of sequences are present many times in a repeated fashion. But each of the repeats, there are spacer elements, spacer DNA in between them, interspaced. These palindromic repeats are interspaced with DNA elements. That's why this, and they're very, very regularly placed. This interspaced DNA is also of a more or less specific size. So this is the regularly interspaced short palindromic repeat that is clustered at a position in the bacterial chromosome. And this is called CRISPR. And there is an associated gene, which is called CRISPR associated Cas, C for CRISPR, S for associated. This gene is present in association to this cluster region of the chromosome. That's why it is called Cas. So this is what this CRISPR-Cas system is. To understand it further, we need to understand how did we reach from this CRISPR to this CRISPR, the RNA-guided molecule. For that, we need to understand a little bit the historical development of this particular topic. So if you go back, it was first in 1987 that the first evidence of this kind of a repeated DNA in bacteria was observed, in Escherichia coli, in fact. And they were not working with this CRISPR at that time. It was nothing known about this. 1987 was too early. In 1977 only, the first DNA sequencing technology was developed by Sangers and also Max and Gilbert, 76, 77, that time. Only 10 years have gone and people have probably sequenced many genes, not that many genes, few genes, maybe. And this gene, IAP, alkaline phosphatase isozyme gene, at the 3' untranslated region of this gene, they were getting a repeated sequence similar to the CRISPR. And they have mentioned in that paper, the Ishino group, that this is a peculiar kind of a sequence that we are getting. We don't know what is the function of this particular repeated element in the DNA. Then again in 1993, in an archaea, in an archaebacteria that, these are extremophiles. This archaebacteria, Helophorax mediterranean, this also had a similar kind of repeated DNA element in its chromosome. Then it was a period of a genome sequence in 1992 to 2002, and it is continuing now that there are whole genome sequences. The sir was mentioning that he has also done a whole genome sequence. So that this was an era when the whole genome sequencing started in 1992, 2000, that period. A lot of bacteria were sequenced. Human genome project were also going on. Drosophila genome project were also going on. And there are a lot of plants, rice genome project, Arabidopsis genome project, Cinorhabdacus elegans genome projects. All those projects started during 1990s, and sequences were available. And when these sequences were compared on with the database, it was found that 40% of all bacteria and almost all archaea have this kind of sequence repeats in their chromosome, in their genome. But what this is doing in the genome, it was not known to scientists. And everybody was surprised that such a sequence is it is universally present in so many different archaea, and I don't know why 60% bacteria are lacking this. This is a question which is hovering in my mind also. But all of the archaea, why do they contain this repeated sequence? It was Janssen in 2002 who first who got this Cas sequence associated with this CRISPR element, and he coined the term CRISPR for the first time in 2002. And then, even then, it was not clear what is the function of this gene. In 2005, 3 back-to-back papers by 3 different groups, by Mojica group, by Bolotin group, and Fausel group, they published 3 papers on different organisms, different bacteria. One of them was Yersinia pestis by Fausel group. They could say that the interspaced DNA in between the repeated elements are of foreign origin. They are not of bacterial origin. They are coming from somewhere else. They have a sequence similarity to phages, bacteriophages and plasmids. So here from the concept arose that probably this CRISPR is a kind of a immunological memory. It is a kind of adaptive immunity that bacteria have stored within them. With the help of these spacer elements, the bacteria, these spacer elements are of foreign origin. They belong to certain phages, which have once infected the particular bacteria. And they have saved that snippet of DNA within their chromosome so that they can recognize whenever the same phage attacks the same bacteria, and they can nullify that phage. So that was the concept growing up in the minds of the scientists, and they were getting similar kind of evidences in various research. It was 2006 only when Doudna, Jennifer Doudna, a very prominent, very, very enthusiastic scientist, you can say, who had dreams in her mind that I had to do something. But she was actually a worker of RNA interference field. Doudna got introduced to this area of CRISPR-Cas9 research. She was brought to this area by one of the geomicrobiologists in UCLA, University of California, Berkeley, where Doudna was appointed that year only. Jill Banfield was searching for an RNAi expert, and she found that someone, Doudna, is there in her own institution, and she called her over phone and discussed about this CRISPR and told her that, do you know about this thing, CRISPR-Cas9? Doudna never heard about this CRISPR, and she heard for the first time. And they, in this coffee house, they were discussing, and they chalked out a collaboration that if we collaborate to learn about this CRISPR-Cas, it will pay something in future. If you want to know more about this, you can go for this book, A Crack in Creation, written by Doudna and Steinberg. So in 2007, the CRISPR system was used for the first time, not for the gene editing purpose, but since it was known that it is CRISPR is an adaptive immunity that is providing immunity to the bacterial cell. And this was a private group, a Domisco, a Danish group, which were, they usually produce yogurt and curd. That yogurt production requires a bacterium called Streptococcus thermophilus. It is fermentating bacteria, and it's very important. But the yogurt products got affected by phage infection at that time. So they had to find some solution to the phage infection. So what they did, they cultured the Streptococcus thermophilus with the phage, and then they plated just to find some phage resistant clones over there. When they plated, they got some resistant clones out of those many bacteria out of 10 to the power 9. They got some 10 or 12 such clones, which they picked up, they converted it to pure culture. And when they sequenced the DNA of this pure culture and this pure culture, there was a difference from the earlier sequence to the new sequence. They got that the cluster area has increased a little bit. A new piece of DNA has been inserted in the the mutant organism that they are getting out of this. So what is this? This bacterium has picked up a small piece of DNA from this phage. So that's why when it was matched with the sequence of the phage, they got that this sequence is matching with the phage DNA sequence. That means they could prove that, okay, phages are incorporating their DNA. They are not incorporating, rather, it will be better to say that the bacteria are taking up a piece of their DNA, and they are saving that DNA in their genome so that they can recognize these phages in future. Just like you can imagine, just like the COVID-19 virus, you are going through all these things. You're being vaccinated nowadays. Why? Because our immune system is also capable of memorizing previous infections. So if you introduce the vaccine is what? The antigen. If you are introducing the antigen in your body and you are asking your body to remember this antigen so that it will prepare the instruments, the tools, those are called lymphocytes, B lymphocytes and T lymphocytes, those are in a different way. And whenever that same antigen in the form of the real virus enters into your body, those lymphocytes will prepare the arsenals, antibodies and the T cell receptors to fight against those freshly coming viruses if it is same. So here was it was proof that this thing is true. The immunity of bacterial immunity goes like this against the phages. And this was the first application that they got a new strain which was resistant to phage, and this problem was removed. But at this point of time, a lot of questions remains to be answered, that how the phage DNA snippet gets incorporated in the bacterial genome, how the CRISPR DNA is transcribed and processed into small pieces of hairpin-looped RNA, and how is the target DNA recognized and is destroyed. All these questions were not answered, even though it was known that it is being the adaptive immunity of bacterial system. So, and finally, how to apply this knowledge to human use? This was the biggest question hovering in the mind of Doudna especially. So the collaboration of Doudna with Jill, as well as with one more scientist, John van der Oost, resulted into two back-to-back publications in 2009 and 2010. And there they could isolate these three genes, the three proteins rather, Cas1, Cas6, and Cas3, which they deciphered that it works like this. This is the CRISPR element in the bacterial chromosome. This is the bacterium. And this is the phage that has infected first. At the stage 1, what happens? The Cas1 enzyme, it cuts the DNA into small pieces, and a small snippet of the DNA is inserted as a spacer. This is called protospacer. It is inserted as a spacer into this clustered CRISPR area. And this virus is now has been remembered in the form of this interspacer DNA element. In the next stage, what happens? A pre-CRISPR RNA, a long RNA molecule is being synthesized of the whole CRISPR sequence. And that is being clipped into small pieces by the Cas6 enzyme like this. And this area, one of them, as you know, it is the whole CRISPR area. One of the molecule will be complementary to the target DNA, the viral DNA. So whenever the viral DNA, the same virus attacks the bacteria, it releases its DNA inside the cell, and the Cas3 enzyme, it comes and it binds with this this molecule, this RNA molecule, and it clips the incoming target DNA into pieces. This is how this immunity is achieved. But the problem with the Doudna's findings were that this Cas3 is associated with about 10 to 12 extra protein molecules. So it was a complex kind of a structure, which was actually disturbing Doudna, that how can we use this complex molecule for maybe gene targeting or for identifying viruses or some other human use. Direct application was difficult with such kind of complex structures. So it was Emmanuel Charpentier who was working far away in France in the similar field with yet another bacterium called Streptococcus pyogenes. Streptococcus pyogenes is a pathogenic, it's a human pathogen. It's called flesh-eating bacteria. It's a very dangerous bacterium with which she and her group was working, and they had published a paper in this prestigious journal of Nature in 2011. And she got a little bit different findings that along with the CRISPR RNA and the Cas enzyme, there is yet another piece of RNA, piece of sequence of DNA that produces an RNA that is called Transcriptor Rna, that is also required for the process of this adaptive immunity, or this interference, you can call. So what happens is, they found that this piece of rest of the part were similar. The only difference was this, that the enzymes required for this process was, the cleavage process was simple. It was a single protein called Cas9 rather than Cas3 and multiple multi-subunit proteinaceous structure. It was simply a single protein structure called Cas9. Till then, Charpentier's group was not able to purify Cas9. She knew that there is a CSN1 gene over here that is producing an enzyme, and that enzyme is causing this degradation of Paj DNA that is entering into the cell. And Transcriptor RNA has to hybridize with the CRISPR RNA first, with the pre-CRISPR RNA first, and that is chewed up into pieces. Up to that, this was quite similar, except that this Transcriptor RNA is an extra molecule that is required. That was what found by this Emmanuel Charpentier's group. So what was next? In 2011, fortunately, Doudna and Charpentier, they met each other in a conference at Puerto Rico, and they went into a collaboration that let's work together and find out what this CSN1 gene is producing, what is the nature of that protein. Doudna's lab was expert of protein isolation, protein structure determination, biochemistry, etc. And Emmanuel's lab was purely on microbiological work. Microbiological expertise was there in her lab. So what Doudna and her group, particularly Martin Genet, is her postdoctoral fellow, what they did, they isolated this Cas9 enzyme, they characterized it, and finally they fused this Transcriptor RNA with the CRISPR RNA and converted it into a single guide RNA. To make it as simple as that you require a single guide RNA and this Cas9 protein to target your DNA. And what is interesting is this, that this 20 nucleotide guide RNA, this position, which will bind with the protospacer DNA, you can program this area. You can use your own desired sequence of nucleotide, ribonucleotide over here, and it will go and bind with the target sequence, and it will introduce a double-strand break. So what after double-strand break is introduced? See how this DNA or RNA molecules are inserted into the cell. You can directly insert the guide RNA along with the Cas9 by microinjection or electroporation, or you can insert that with the help of plasmid DNA, extra chromosomal plasmid DNA that will contain those genes, the single guide RNA gene as well as the Cas9 gene. So they will express in the cell, and they will then make this complex, single guide RNA Cas9 complex, and will target the particular DNA with which this blue area, the programmable area of the RNA hybridizes with. So either of the strategies you can use. This is when you are using this one, the RNA is less stable in the cell, so you have to study very quickly, and this one is a stable system. So when you introduce this, and what happens, a double-strand break is created, and the cell wants to repair that break. So when the cell wants to repair, it introduces mutations. Either it deletes some nucleotides at that position, or it inserts some nucleotides at that position, or it substitutes some of the nucleotides like A to A, T to G, C, like that. So these kind of mutations are always causing, will create a knockout mutant or null mutant of that organism. That organism will not be able to produce the gene from this particular area because that is the mutant version, or the particular target DNA can be converted, can be corrected if there is some certain error, like there are different diseases in human beings which are because of the sequence errors. So you can rectify those errors if you introduce a donor DNA as a homologous DNA. So homologous direct repair system will repair the double-strand break like that. This is called non-homologous end joining. This is called homology-directed repair system. So either of this system will be used to get your result done. So now I'm going to talk about the application part. Okay, desirable characters for applications that we need to feed the ever-growing population are faster growth, higher yield, disease resistance, stress tolerance, and alteration in the taste, texture, and nutritional value of the crops. But the conventional methods that are available are selection, hybridization, heterosis, mutation breeding, and others. They are very, I mean, they're there, but there are some problems with them. They are slow and time consuming. The problem of linkage is there. That means when you are trying to introduce one character, the other character automatically enters into the target organism, into the crop. Lack of purity and termination of sudden drop in the yield. Termination or sudden drop in the yield also occurs. Suddenly you are getting a high yielding variety. In the next generation, you might get a low yielding variety out of that because of the variability or termination problem. Modern methods of transgenesis developed in the last decade, last decades of the last century. In fact, there already 40 decades have passed when this transgenesis is going on parallelly. It is also a time consuming. In transgenesis, what you have to do, you have to isolate your DNA from one organism, then you have to clone it into a certain vector, then you have to transfer it to your target organism. So it is a little bit longer. There are many steps in that. But in case of gene editing, you are directly modifying your target organism. The isolating genes from other organisms is not required for this purpose. So it is less time consuming, and it is the homogeneous horizontal gene transfer problem is also avoided. Environmental issues are also less important when you are directly modifying a certain gene transfer. Horizontal gene transfer problem is removed. That's why the environmental issue is also getting less significance. So some success stories regarding this, I will go very fast in this area. This was done, the mutation in FAD gene in soybean to improve oil quality. This was some users of gene targeting. I'm talking about CRISPR-Cas9 use. In this case, FAD is a gene, fatty acid desaturase, that converts oleic acid into linoleic acid. Linoleic acid is harmful. It is pro-inflammatory and thrombogenic. So if you mutate this gene, oleic acid will not be converted into linoleic acid, and you will get more amount of oleic acid in your seed, and that is beneficial. So this was the first mutation. I will not say first. There are many. I have chosen only a few as an example. This is low gluten wheat. Can it be removed? This low gluten wheat removal of anti-metabolites. This was the next thing that you can concentrate. Low gluten is required. Gluten is a problem in some individuals, particularly in Western countries. A disease called celiac disease is caused because of the gluten. Gluten is made up of two proteins, two polypeptides. One is called gliadin, another is called glutenin. So scientists modified this gliadin protein, this alpha gliadin. You can see this is the control lane, and these two are mutant lanes, where this alpha gliadin is absent. And they proved this also via this mass spectroscopy, MALDI-TOF rather. And you can see the interesting thing is this, in gene editing, what happens, this is the wild type organism, and this is the mutant organism. This is wild type, this is mutant. This is wild type, this is mutant. You cannot distinguish by morphologically the wild type from the mutant. This is so specific that the gene that you want to disturb or to disrupt, you simply disturb that. So linkage problem is not there. And the removal of toxin in cassava. Cassava contains a toxin, glycogen, cyanogenic glycoside, and because of a certain gene, and that gene was mutated by CRISPR-Cas9. This cyanogen glycoside is very significant in amount, and if you consume it regularly with low protein diet, it develops into a disease called Conso disease, and it's a common disease in African countries. So by converting this to the low toxin cassava was an important development in by this CRISPR-Cas, using this CRISPR-Cas technology. This is the blight-resistant rice variety that was developed by this CRISPR-Cas9 system. As you know, the bacteria, the Xanthomonas oryzae and other bacteria, what they do, they deliver a protein called TALE. This is a transcription activator-like element, which goes and binds with genes in the plant cell, and those genes are overexpressed for the and the product of those genes are beneficial for the bacterial growth. This is how bacteria utilize the host cell for their own growth. So what scientists did, they modified a gene called sweet gene. Sweet gene is required for the transport of sucrose from the cells to the from the source cell to the sink cell. Sweet is for sucrose will eventually be exported transporter. Sweet stands for that. So what scientists did, they mutated the sweet gene by using CRISPR-Cas9 so that less amount of sucrose will be available for the bacteria to grow in the extracellular spaces. So with these 4 examples and this list, there are a lot of, in case of rice, there are a number of target genes that have been modified using this CRISPR-Cas9 system. Some of them use some of them use non-homologous end joining method. Some of them use homologous repair method. By that method, they have modified several different genes in rice plants. And in India also, you see several different labs are engaged in CRISPR-Cas9 research. I'm not going in details in each of them, but a few of them, like Bose Institute, is also doing developing an optimized toolkit for inducible genome editing and regulation of gene expression in tomato plant. ICAR, IARI, ICGEB. So there are many different labs that are engaged in this kind of CRISPR-Cas9 research. I will not lengthen my talk. It is already delayed. 3 minutes, I'm over. So thank you very much for your attention.
Thank you so much, sir. This was really an enriching session. So, sir, there are a few questions in the chat box. So I read a few of them for you.
Sure.
So first question from Devananda Banerjee. Does Cas9 cause double digestion if there are very similar DNA sequences in genome with only few differences of nucleotide?
Yeah, if you have very similar, yeah. One thing is this, in fact, you can use many different guide RNAs at a time. More than, even you can use 10 different guide RNAs at a single experiment, single shot, you can say that you can convert or mutate 10 different sequences in a cell, in an organism, in one shot by using those guide RNAs. And if, of course, if it matches, if 90% of it matches with a particular sequence, then there will be problems. So when you design your, that 20 nucleotide sequence in the guide RNA, you have to keep that in mind also, of course.
Thank you, sir. Sir, next question from Kollan Mohapatro. Can organellar genomes, that is plastid or mitochondrial genome, be transformed using CRISPR-Cas9?
You can do that. Chloroplast genome has been targeted by this method. I did not bring that in this lecture. But you have to use protoplast technology, and you have to screen for that after you electroporate the DNA or the RNA, and you have to select the mutant in which you get the proper result. If it is a chloroplast genome, yeah, of course you can do that. Even in mitochondrial genome, you can make some changes using this, but a little bit difficult than the nuclear genome, of course.
Thank you, sir. We have to end the discussion here. Anybody have any queries, please email, sir.
Of course, yeah. Welcome.
Thank you, sir. We had a great time listening to you. So now I request my dear friend and colleague Annisha to host the next session. Over to you, Annisha.
Thank you, Kosturi. Thank you, sir. Now our next speaker, Mr. Uday Shankar Rai, have with us. He is an agricultural professional having more than 12 years of experience in this field. After completion of MSc in plant breeding from Bidhan Chandra Krishi Vishwavidyalaya, he started his journey as an official for Maharashtra State Seed Corporation Limited, which is undertaken by Government of Maharashtra, a leading public sector seed industries in Akola, Maharashtra. After that, he had changed his career to purely research-oriented field and achieved diversified research experience in ICAR CRIJAF as Senior Research Fellow in different important projects during 2011 to 14. And also worked under WBADMIP, that is World Bank Project, as Agriculture Specialist during 2014 to 16. Presently, he is working under West Bengal Agricultural Services and posted as the Assistant Botanist and in charge, Zonal Drought Resistant Paddy Research Station, Hathwada, Purulia. He was associated with AICRIP, that is All India Coordinated Rice Improvement Project, and number of different projects in International Rice Research Institute. He has developed a number of advanced rice line suitable mainly for upland rainfed ecology with other desirable characteristics. Among those advanced lines, 6 number of lines are at the age of the pipeline and expected to release as variety during 2022 to 24. He has published a number of research publication and different articles in national and international journals, and a number of leaflets, booklets, popular articles in English and Bengali language for enriching the farming community. Recently, in June 21, an article has published in Scientific Reports where he is a co-author. With this brief introduction, I would like to call upon our next speaker, Mr. Uday Shankar Rai, and his topic for today's webinar is Present Scenario, Challenges and Advancement in Rice Development. So please take the screen.
Thank you. Namaskar. Am I audible?
Yes, sir.
Yes, sir. Yes, sir. You are audible. Clearly audible.
Good afternoon. It's my great pleasure to be here with you in this great platform. And my sincere gratitude to Principal Sir and President Dr. Mandal Sir, Professor Mandal Sir, and Dr. Pandey Sir and Dr. Baramani. Now, it's a tea time. Though it's a tea time, I am sharing you good dishes of lunch. Hope that all we have taken lunch, and most of us taken lunch we have taken rice in lunch, and not only we, half of the world population depend on the rice for their food security. And in India, rice is a staple food and key of food security. And here, my video is off probably. Hello. Okay. Here, the rice producing area. Show the map. I have just the color, green color, are the rice producing area. Asia contributes significantly with 90% of global rice production and its consumption. And the deeper color shows the higher production area. So in Asia, the leading rice producing country are India, China, India, Indonesia, Bangladesh, Vietnam, Myanmar, and Thailand, Philippines. And in Latin America, other than the Asia, there is Latin America and Sub-Saharan Africa is producing. And if we see the table, it shows that the India contribute 21% of global production, and West Bengal contributes 15% of our national production. And the productivity is comparatively lower. Our national level productivity is comparatively lower than global productivity, that is 4417 kg per hectares. And West Bengal is, to some extent, it is higher than our national level production level, that is 2730 kg per hectare. And our area is comprises in West Bengal, we have put the area both for Kharif, Boro, and Amon, Aus, and Boro, 3 seasons, combining the 3 seasons. And our national demand of rice on 2050. And Indian population is increasing and estimated to be 1.6 billion by 2050. So to feed this higher population, we have need to produce 137 million tons of rice annually in 2050. And where present production is, in my previous slide, you have looked at 104. So we need a great effort to change and to achieve this higher production. Here is the institute. I have put this institute. Other than the university, agriculture university, and research organization, this institute are mandate work for their rice development. In international level, it is International Rice Research Institute. This is in Philippines, and it is a substation for Southeast Asia in Varanasi. And in national level, there is 2 premier institute. There is National Rice Research Institute at Cuttack, Odisha, and Indian Institute of Rice Research in Hyderabad. And in our state level, one institute is there, Rice Research Station, Chinsura, we all know that. And it has 2 substations, one is for drought and one is for salinity and flood. And now come to the rice genome. Oryza genus is thought to have originated about 14 billion years ago in Southeast Asia and Philippines. Since there it is dispersed and distributed throughout the tropic, and there is 2 species, cultivable species, Oryza sativa and Oryza glaberrima. Oryza sativa is popularly known as Asian rice in grown worldwide, and it is first domesticated from Oryza rubipogon. And it has 40,000 of variety, and it is grouped in 4 groups: Japonica, Javanica, and Indica, and one is aromatic, I have not put here, and Japonica. First of all, Japonica. Japonica is a group of rice of Eastern China, northern and Eastern China, and it is characterized by short plant and grain short and round and sticky when cooked. And Javanica is popularly known as tropical Japonica. It is a rice of high altitude. It cultivated in the rice terrace of Philippines, and it is also characterized by tall and light green leaf, and grain is long and bread, sticky when rice. And Indica is the Indica is the major type of the rice grown in subtropic, tropic, and subtropic. It is characterized by long plant height and non-sticky when cooked. And glaberrima is African rice, and it is cultivated in degraded soil, and it is a very good source of useful genes, that is nutrient efficiency and biotic and abiotic traces. So, and here is a variety. Just I put a few photographs of variety because we are concerned about varietal development of rice. And here we all know that it is the last last rank of lowest rank of the taxon. And here is a few variety developed by our rice research station. And here our development from since 1911 to present area, I have divided it in four segments. Here you look, there is 3 segments and another in next slide. Here I have first segment is from before 1950. Rice breeding was first started as GP Hector as an economic botanist in 1911, and during this period, major natural screening, natural selection was exploited hugely, and 445 varieties developed. And during this period, research organization are establishing here. Our Indian Council of Agriculture Research started in 1920, and at that time, the Bengal and Madras was the only province which was specialist exclusively of rice crop. Hence, the Rice Research Station Chinsura has established in the 1930. It's a too old research station in our state. And then India National CRRI was established, which is National Rice Research Institute now. And at that time, only 445 variety developed. And from 1950 to 80, this is the most important time, and it is during this time, the screening of exotic type of genetic stock, systematic development was taken place. And first, there is inter-racial hybridization program between Japonica and Indica was initiated during 1950 and 54. But from this program, only 4 variety was developed. But All India Coordinated Rice Improvement Project was started from 1965, and interracial hybridization program between semi-dwarf, Taiwanese type, and Indica type started. And from this program, a remarkable output have come, and our Taiwan TN1 gene and our variety Podda Joya and IR8 miracle rice was evolved from this program, and this is the responsible for our green revolution in rice. Now, in third phases, 1980 to 2000, it could be says this is the era of hybrid rice. Though hybrid rice is not so popular in our country, but it is only have 2.7 to 3 million hectare area, and only 91 hybrid rice was released. And by using cytoplasmic male sterility, commercial rice hybrid rice production was started. But for some reason, hybrid rice is not getting popular in our country as well as in our state also. And third one, and what our previous lecturer, Dr. Pandey sir, has elaborately tell you, this is the session of genetic, this is a biotechnology era. In this session, there is genomic editing, and also there is some we are able to develop some important thing, and yet to harvest and more thing from this genomic editing. So one thing I would like to mention, green super rice is now in field in our India. So for production of it is using the for our low nutrient capacity, high resistance for biotic and abiotic traces, and high grain yielding. We are also testing this green super rice in our research station, and it is performing, and it could be the next one jump to our rice production. And now another will be by gene modifying the gene editing program in futures. And here is the challenges for our varietal development. As land scenario, we all know that by existing population, increasing population, the per capita land is decreasing. So within a small land, we have to produce our 137 million tons of rice within 2050. So land is a land area decrease is main challenge. Climate challenges, now frequent drought, cyclone and flood, enhancing temperature and increased CO2, and water related concerns also there. Water availability and water quality. Water availability, we all know, some of us know that for production of 1 kg of boro rice, presently we have to spend 3,500 liter to 4,500 liter of water for producing 1 kg of rice. So this say we are using lots of water for rice. So rice, water efficient rice is very we are demanding thing. Now biotic traces and about here is biotic trace. On first author, Dr. Professor Mondal has said very elaborately the disease resistant and pest resistant. But I would like to just I would like to show you how the pest scenario is changing. In 1965, only 3 pests are there: Eurostem borer and green leafhopper. And now in 2015, it is 19 pests are there, and we have to tackle 19 pests for getting a proper yield. And on the other hand, disease. On the other hand, there is also one on 1945. There was only brown spot disease was there reported, but now it is also increasing very rapidly. So it is also a challenge to us. And nowadays, to feed our poor lower group in peoples through rice, it is very essential to enrich rice with vitamin A, zinc and iron. And zinc is very important. We have just realized this during COVID, and some of us also taken zinc tablets also. So zinc enriched rice also a priority area. Now the ecosystem. Rice is diversely adapted to ecosystem, but every variety is different. Irrigated ecosystem, there is another variety. For rain-fed ecosystem, there is another variety, and for low deep water rice, there is another variety. So for all ecosystem, here we can see that the irrigated ecosystem is the higher areas and production is higher from irrigated ecosystem, but other ecosystem also remarkable area and production. So for all the ecosystem, we have to develop a separate variety. And now here I have just showing you the few varieties developed from rice research station Chinsura during 2017 to 2019. Chinsura Nona 2, it is a salt tolerant, salinity tolerance variety. And now Muktosri is a low gain arsenic accumulator variety, and Bhupes is a very high yielding variety. These are we are now developing this variety for different ecology and for different characteristics. And now thank you, and to keep our effort to keep our food security, এবং সে আমার সন্তান যেন থাকে দুধে ভাতে, এইটা আমাদেরকে বরাবর রেখে যেতে হবে, এটাই আশা। থ্যাংক ইউ।
Thank you, sir, for such an informative and enriching session. It will surely help us a lot. Now I am moving to the interactive session. We have some questions over the chat box. If you permit, can I take the question, sir?
Sure.
From Debanando Banerjee, question is there any change in physiology of crops due to aerobic method of rice cultivation?
Actually, aerobic method of rice cultivation, for aerobic rice, there is a separate group of rice. That's a aerobic rice.
Okay.
Hello.
Yes, yes. You can continue.
Is there is a change in physiology of crop due to actually there is a separate group of rice variety for aerobic condition, and they are not waterlogged condition. They grow in aerobic condition.
Okay. Another question from him. What are the effects of boron application on rice growth and development?
Oh, very good. We are conducting a trial now. We are using a zinc, boron, and urea. It is in increasing the yield and the taste to it also, grain taste to it also. Our field. It's possible to cultivate rice in saline soil.
Okay, sir. Another.
Next question is: it is possible to
okay.
cultivate rice in saline soil? Yes, there is possibility, and the farmer are cultivating in saline soil in rice. If you go to the Goshaba Sundarban area, and after that I have just mentioned Chinsura Nona 2 IET number. It is our recent release variety for salinity condition, and it is the salinity tolerance. It is up to EC6 decimal per DS per M. Okay. And another is zero tillage cultivation in West Bengal. There is a zero tillage. Nowadays, we are giving importance to the direct seeding rice, and zero tillage rice is there is in Bardhaman Ausgam block. There is going on, and in North Bengal, some portion there is presently zero tillage through seed drill, seed and fertilizer mixture. Driller, and another direct seeding through zero tillage and through drum seeder. We are giving important, where for the rain-fed area we are advocating them to go to direct seeding by through zero tillage machine, and for irrigated area there is for through drum seeder. Any more?
Sir, now we are running
Okay,
very short
okay,
in time.
thank you.
So thank you once again. And for the students, you can mail your questions to the speaker's mail ID. If you have more questions, you can mail them. So I think no more questions are there. Thank you once again. Now I hand over the responsibility to conduct our last session of today's event to my dear friend Kosturi. Over to you, Kosturi, please.
Thank you, Anisha. Thank you, sir. So before I welcoming our next speaker, I have an announcement for all concerned. The participants are requested to fill the feedback form for the generation of certificate. The link is given in the chat box. So now I would like to introduce today's last speaker, Dr. Krishnendu Pramanik, UGC Dr. D.A. Kotari Fellow of the Department of Botany, Vishwabharati, under the guidance of Professor Narayan Chandra Mandal, who has already enchanted us with his wonderful speech today. Dr. Pramanik has completed his BSc and MSc from the University of Bardhaman and was a gold medalist in his master's. He did his PhD from the same university under the supervision of Professor Tushar Kanti Maitri. His research career is mainly focused on plant microbe interaction, environment microbiology, bioremediation, and computational biology. Currently, he has been working on the plant and bacterial transcriptomics under condition. He has published more than 20 research papers and book chapters in his 7 years of research career. At present, he is serving as a guest associate editor and topic editor of Frontiers in Sustainable Food System and topic editor of Frontiers in Microbiology. He has also worked as a reviewer of various well-known journals. So today his topic is phytobeneficial rhizobacteria, a boon for the bane of heavy metalloid contamination in crops. Now I request him to deliver his talk. Over to you, sir.
Thank you, Kasturi, for this nice introduction. Am I audible as well as visible?
Yeah, yes.
Okay. Can I share my slide now?
Yes, sir.
Okay. Thank you. Is my screen visible?
Yes. Pointed is also visible.
Okay. Thank you. Thank you, Kasturi, once again for your nice introduction. So, I am Krishnendra Pramanik. I will be present here. Just 1 minute. Just 1 minute. There is a technical problem. Is the slide visible?
Yes.
Okay. I am presenting here my topic, phytobeneficial. What is going on? Please allow me 1 minute. There is a problem. Hello. এটা স্ক্রিন আসছে সামনে একটা স্ক্রিন চলে আসছে Kasturi, I am leaving and rejoining. Okay, there is a problem. Okay.
No.
কৃষ্ণেন্দুর কি প্রবলেম হলো গো তোমার?
তো জয়েন আবার মনে জয়েন করতে পারছি না, আমি কি জিজ্ঞেস করবো না?
Hello.
এই তো
So I am here. Okay. There was a problem. Okay. Am I audible?
You are audible. Now you start.
Okay. Okay. So my topic is phytobeneficial rhizobacteria, a boon for the bane of heavy metalloid contamination in crops. I have prepared my slides especially for the students who may get some idea about what I'm working now. I'm just a beginner and I'm a budding researcher, just doing my research on PGPR or phytobeneficial rhizobacteria. Okay, so I'll be describing about the problems, the possible solutions, and the acts that are being done in both laboratory and in both field condition. All of you know that India is second largest populated country in the world, and the rapid urbanization and industrialization leads to gradual loss of agricultural lands. You can see the paper. Here is a paper, Pandey and Seto. This is from Journal of Environmental Management, published in 2015. Although it is a 6 years back report, but still it is relevant that there are loss of agricultural lands over the year. Sorry. Pandey and Seto have combined remote sensing data with the agricultural census data and compared to give us some idea about the loss of agricultural lands. This is the moderate resolution image spectroradiometer, or called MODIS. This is a satellite which captures images of the various types of images from over the globe. And if you can see in India, that Maharashtra, Chhattisgarh, Madhya Pradesh, even in West Bengal and Uttar Pradesh, is gradually losing several hectares of agricultural lands over the year. And if you can see, especially in West Bengal in Bardhaman, in the southern part of the Bengal, there is a gradual loss of agricultural lands. So for the sake of students only, I am sharing this screen that what are the heavy metals and metalloids. All of you know that, but some students may or may not be acquainted with these. So what are the heavy metals? Heavy metals are actually the metals with a density above 5 gram per cubic centimeter. These are some heavy metals: cadmium, chromium, cobalt, and metalloid such as arsenic, antimony, etc. So there are some permissible limits of different types of heavy metals according to WHO or other Ministry of Environment, etc. So why the heavy metals are toxic? Because due to the strong affinity with the metal cations or sulfur and the sulfhydric groups in many enzymes which react with the ingested metal cations, which can deactivate the enzyme, which stops or alters the metabolic process, even lead to cell death. So you can see the cationic forms of the heavy metals. But in case of mercury is toxic even at its elemental form. These are some heavy metal phytotoxicity, which ultimately leads to poor growth, poor yield, increased heavy metals in grains. So the food chain contamination occurs both through direct and indirect mode. Okay. There are several types of toxic effects in human also. Ethylene disease is caused due to cadmium poisoning. Arsenic causes skin lesions. Mercury poisoning leads to several types of diseases like Minamata disease, pink disease, etc. And lead poisoning also cause severe symptoms in the humans. We have to understand that plants are immobile in nature. Plants have to stay within where they are. Plants cannot move. Plant cannot escape. So there may be several types of environmental stress, both biotic and abiotic stress, but plants have to stay there. But up to a certain concentration of heavy metals, plants reduce their growth, even they die. But some are of their strains, that is heavy metal resistant strains, they are helping help the plants, and some heavy metal sensitive bacteria also, they can dry or escape from this environment. So our strategy as a role of microbiologist is PGPR assisted bioremediation to develop eco-friendly, cost-effective and sustainable approach to check the heavy metal immobilization, creating awareness to the farmers, to control or avoid the chemical fertilizers, use of PGPR based biofertilizer, and maybe GM crops may be developed. So how we can approach about this? We can isolate native or indigenous multi-metalist and PGPR for the bioremediation and plant growth promotion purpose. And first we have to check in the laboratory. After that we will go to the field and observe whether there is an improvement of grain quality or quantity. and finally to prepare the PGPR to the market for easy accession of the farmers. So the term is plant growth promoting rhizobacteria. Before going to rhizobacteria, we have to understand some types of roots. All of us know that there are several botanists here also. So there are some taproot system and there are some fibrous root system. Initially, we think that there is the soil adhering to the root particles is rhizoplane soil. But the concept is somewhat modified in 2013, which is published in Nature Education. 13. The term first, the rhizosphere term was first coined by Lawrence Hiltner, who described that to describe the plant root surface, and the plant root surface that is inhabited by a unique population of microorganisms influenced by the chemical exudates or root exudates. He classified the total rhizospheric zone into 3 parts. First one is endorhizosphere, second one is rhizoplane, and third one is ectorhizosphere. There are 2 types of concepts. First one is extracellular PGPR or ePGPR, and maybe there intracellular PGPR or iPGPR strains. So the concept has somewhat been modified. These are the scientists or the key persons who discovered first, who termed the point first, the plant growth promoting rhizobacteria. Here, the paper, the original paper, Pfeiffer was the assistant professor in the University of California, Berkeley, and now is an emeritus professor. And Sprott was also a plant pathologist in University of California, Berkeley, and now he is also acting as emeritus professor in Department of Environmental Science and Policy Management, UC Berkeley. So what are the PGPR? The PGPR, the microbial communities grown in rhizosphere, which are beneficial for plant growth, yield and crop quality have been called as plant growth promoting rhizobacteria. That is also given by the definition given by Cooper and Sprott. PGPR have to fulfill some criteria to be named as them as PGPR. The term are biofertilizers, phytostimulators, rhizoremediators, biopesticides, and biocontrol agents. There are 5 main points. PGPR should at least possess 2 to 3 characteristics to be named as PGPR. So what are the heavy metal resistant PGPR? So unlike any metal sensitive bacteria or PGPR, heavy metal resistant PGPR can resist or detoxify the toxic effect of 1 to several heavy metalloids by active or passive mechanisms and play a dual role of heavy metal resistance as well as plant growth promotion. So why we are opting the heavy metal resistant PGPR to be used in the field condition? So the main purpose is it's eco-friendly, it's inexpensive or less expensive, it's effective, and it's sustainable. So what is meant by sustainable agriculture? This is very important to maintain in the field condition. That is an integrated system of plant and animal production practices have a site-specific application that will last over the long term. This is a concept. The concept was already there, but we recently published a book chapter where we have given that the two types of thoughts. One is bacteria-assisted phytoremediation, and another one is PGPR-mediated bioremediation of heavy metals. So rhizospheric microorganisms there, they may be resistant or maybe susceptible. We have introduced that bacteria-assisted PGPR-mediated bioremediation that prevents heavy metals to flow to the aerial parts of the plants, because the grains will contain the lower contents of the heavy metals. So heavy metal immobilization is crucial for any types of PGPR implementation in any types of soil or agricultural soil. So why the bacteria or PGPR are colonized around the roots? Do they get something? Yes, they get something that is carbohydrates. Where from? From plants. Plants gives them carbohydrates, sterols, amino acids, fatty acids, enzymes, flavones, lignins, various types of indole compounds, and several types of organic compounds, and they act as mutualistic relationship, and they used to produce some bacterial phytohormones and help plants to grow better. So here is the simplest form of the plant metal microbe interaction given by Sharma and Archana 2016. So what here the cadmium is represented here as a representative of the heavy metals. What they do? Bacteria or PGPR induce cadmium-induced hydrophore. They solubilize phosphate. They colonize around the roots. They produce acetodeaminase. Some of them, of course, they produce phytohormones, antioxidant enzymes, etc. And the effect of which is increased nutrient uptake, reduced oxidative stress, increased heavy metal tolerance, reduced cadmium content in floods. That is the ultimate goal of us, because the slow poisoning matters. We are consciously or unconsciously consume some foods that we have given a slide initially, that we are consciously or unconsciously taking some foods that contains the heavy metals. We should reduce them. So how the bacteria manage? Bacteria can manage it. Bacteria manage by different types of adopting different types of survival strategies. First one is bioaccumulation. Second one is microbially enhanced chemisorption of metals, biomineralization, biotransformation, bioleaching. There are several types of strategies they adopt depending upon the strains, various strains of the microorganisms. Next one is, hello. Next one is, these are some families. These are some families of studied agricultural crops from where the bacteria or PGPR have been isolated. You can see that Poaceae is the dominant family from where the heavy metal listed PGPR has been isolated. We have actually in this book chapter collected the data of 20 years and 10 heavy metals and represented here. So this will be some informative chapter for the students, especially or any beginner in this field. Here I presented the diversity, abundance, and distribution of heavy metals. You can see here that the PGPR isolated belongs to some different phylums, Proteobacteria, Firmicutes, Actinobacteria, among which the cadmium, most of the bacteria are cadmium resistant or lead resistant, and arsenic resistant, and mercury resistant PGPR are less in number because of its high toxicity. So next one is PGPR produce several types of, I've already said that PGPR produce several types of plant growth promoting characteristics like an ammonia production, nitrogen fixation, Asian production. So you can see that sidral production and IA production and phosphorylation and SSCD activity is dominant traits found in most of the bacteria. And one of the important criteria to become a PGPR is biocontrol, which Professor Mondal has said that biocontrol potential PGPR with high biocontrol efficiency controls the biotic stress in fields, apart from the heavy metals, which is a abiotic stress. So we have published this paper along with Sandipanda, Devasmita, Kunal, Sir, and Naranda in Springer. So this is my journey, actually. This is my journey. I am just started my research works in search of heavy metal resistant phytobeneficial bacteria. We first published, this is actually the publication history. We have first published in 2016 the chromium resistant bacteria, which have hexavalent chromium reducing capability, that is named as Raoul Telespi and published in Taylor and Francis. Next, in 2017, we have published in Environmental Science and Policy Research Journal a cadmium-resistant Klebsiella pneumoniae strain. This strain, we have reported for the first time the acetodeaminase enzyme experimentally in this strain. Next, we have collected some and isolated several types of bacteria, cadmium-resistant bacteria, Enterobacter aerogenes, Enterobacter species, Bacillus argyropatai, arsenic-resistant strain, Klebsiella michiganensis, another cadmium-resistant strain. And in 2021, two strains. One is arsenic-resistant Pantua dispersa. Next one is Pseudomonas sp strain. It is published recently from my postdoc laboratory, and I have miles to go, of course. This is the basics of my doctoral research. I have collected some rhizosporic soil samples near the industrial bales and the rhizosporic soil samples was from rice. I have serial diluted it, and this is process. I have screened them, identified them, and checked their heavy metal accumulation potential. And finally, we have checked their efficiency in laboratory condition. We have obtained that a significant reduction of the oxidative stress and increased cadmium tolerance and reduced cadmium content. Germination percentage was also increased, and overall seedling growth promotion was also done. But the circle is, you can see that the circle is incomplete here. We should complete the circle. How it will be completed? We should implement this PGPR to the field, but this is not so easy to apply the PGPR in the field condition. There are several steps. I'm going in my next slide. So how we can apply the PGPR in the field condition. I have started my work in with three different industries: Indian Iron and Steel Company, you know all, ISCO, Durgapur Steel Plant, Durgapur, Alloy Steel Plant, Durgapur. I have collected soil samples, rhizosporic soil samples from there. You can see that there is a crop residue after cutting the rice. There is a crop residue, or maybe it's called stubble. I have collected some samples from the stubbles, and initially I have observed, I have obtained a total of 74 heavy metal resistant PGPR strains. And after a rigorous screening, I have selected CRS2, that is chromium resistant PGPR. Tautel I have already said. And next bacteria are K2, K5, K6, and again I have worked also with K32. So this is the CRS2 strain grown in different concentration of of chromium, and chromium reduction was proven in our research. The CRS2 have some also some PGP traits also. Now I have screened the cadmium resistant strains. These I have established these strains at cadmium resistant, but they have multiple heavy metal resistant potential. I have not seen any data there, because I want to give some precise explanation here. So there is cadmium resistance depending upon various concentration. I have rated and seen there are some rigorous growth in cadmium contaminated plates. Here are the transmission electron microscopic data that indicates that K5 is my Klebsiella pneumoniae strain, which collects or accumulates the heavy metals. This experiment is actually with cadmium, which accumulates the cadmium in the cellular envelope. And my next strain is K6, which is Enterobacter erogens. You can see here also the cadmium peaks, but in control there is no cadmium peak. In case of K6, the cadmium is accumulated within the cell. Okay, then I have characterized some plant growth promoting characteristic, phosphate solubilization, iron production, nitrogen fixation, ACC deaminase activity, which have Professor Mondal already said. This is a really very important trait for being a PGPR, and so ACN production and the plate of hydrophore production. Finally, we have gone through the plant growth promotion study in vitro. Okay, you can see that the DC50 indicates that there is a heavy metal in this case also, but in this case K5 strain is also introduced here. You can see the difference. This is control. You can see the difference because the germination percentage was massively hampered by the cadmium. Here in this case, you can see the tuft of root growth as well as the shoot growth. So the external manifestation was excellent in case of the K5 treated seedlings, rice seedlings. This is trained as rice seedlings, and we have measured the parameters, various parameters. You can see here I have enlarged the views. You can see there are tufts of roots maybe due to some stress. We have to examine what are the factors, what are the genetic determinants, which are the reasons for this, especially for this phenotype. I have gone through some biochemical properties of rice plants, and one of the important experiment was with ethylene. Glick was the pioneer scientist in space biology. Glick first introduced coined the term the space ethylene. You know that ethylene is a gaseous phytohormone, so it is normally available to in the plants. But in case of several, due to the presence of several types of biotic and abiotic space, the ethylene peak has been increased, and that hampers the plant growth. Okay, you can see the peaks. Glick has demonstrated in microbiological research in 2014 that bacteria with SCG deaminase can promote plant growth and help to feed the world. You can see that bacteria, how bacteria perform this? You know that ACC, the substrate, ACC, that is 1 amino cyclopropane, 1 carboxylic acid, or which is the immediate precursor of ethylene. ACC is cleaved by the PGPR association, the intracellular enzyme ACC deaminase, which cleaves the ACC into ammonia and alpha ketobutyrate. So the ethylene production was checked. So, and the growth of plant has been maintained. Next one is which was published in Chemosphere recently. We have worked with some excellent collaborators, and you can see here that the number of you can see the excellent zone of inhibition by the bacteria of some plant pathogenic fungus like Lidosporium, Rhizopas, Alternaria, etc. Here, I have presented that K32 cells is accumulating cadmium also, and here is the root colonization property. Root colonization is a compulsory criteria to be a PGPR. Root colonization is a really a compulsory criteria to be a PGPR. Apart from all these, because if the bacteria cannot colonize around the plant roots, how they will help. So we have checked by fluorescence microscopy how the bacteria helping the plants, and we have seen some bacterial cells around the surface, not within the cells, around the surfaces, using the acridine orange treatment. Next one is the important and the final foremost important criteria, that is we have initially described that cadmium content or heavy metal content should be lowered in crops. We have seen that a significant amount of heavy metal has been decreased by PGPR inoculation. The evidence has been also validated by the XRF analysis. You can see this is a control. There is no detectable peak. This is a high peak in case of EC50 concentration of heavy metal, and this is a low cadmium peak area in case of PGPR inoculated set. It should be optimized. You can see that there is still heavy metals in plant cells, but our aim will be to reduce more and more heavy metals in the plant cells, so that we cannot get the slow poison like the heavy metals. Okay. So we have published these data for the students. I have to say that there are different types of journals, national, maybe national or international. We have to publish, we have to validate what we have done in the research as a researcher. We have to recognize by the world of world scientists or researchers. We have published these papers. This is the most recent publication of us, and this is a review of by Sayanto Mundal with first authorship, and I have helped him to some extent. So, what I want to say that we have completed just the 2 steps of these 8 steps, just 2 steps. I've already stated that I am the beginner. So, from rhizosphere, we have experimented the in laboratory condition, but we have to go the miles to go. There are several steps to formularize or for product development. There are some government regulatory protocols which we have to maintain, and finally to marketize the product as a biofertilizer. So you can see that there are biofertilizers already there in market, commercial biofertilizer. I am proud to share the slides that in India, AgriLife, Nitroflix, Azospidum, Symbion N, etc. are some manufactured from India that are commercial biofertilizer. There are several constraints. We have to face several constraints to implement this PGPR in the field condition. This is really too tough to implement in the field condition. What are the factors? First factor is soil types. Next factor is soil parameters. Next one is biotic interactions. Other soil contaminants. Soil is not about only with heavy metals. There are several types of contaminants in there. So we have to consider several types of other contaminants before application. We have to check the fields, which contaminants are there, which are the nutrient availability, and the loss of genes is the one of the basic criteria. Apart from these work, omics approaches, you should have heard this about, and already Sanjeev Da had taught us about the CRISPR-Cas9. There are some omics approaches, that is genomics, transcriptomics, proteomics, metabolomics, etc. There is a very good focused review by Malik et al. I have just read for this webinar for that the implementation of genetic engineering novel omics approach to enhance bioremediation. This is a very good paper, you can read out. So apart from these, bioinformatics bioinformatics is another omics approach that incorporates computer science, chemistry, biochemistry, biology, statistics, the whole department of the science. It includes bioinformatics. So bioinformatics can be utilized for all researchers to strengthen their work. Here is another paper I can share, that omic technology. This is not a paper, sorry, this is a book chapter, omic technologies for the abiotic stress tolerance in plants, current status and prospects. It is published in 2019. What I am doing currently, that is working with transcriptomics, microbial transcriptomics during plant microbe interaction and under cadmium stress. But you know that due to COVID, the data is still not yet satisfactory for us, so that we can share for you. However, I am also interested in bioinformatics also. I have I have studied some microbial proteins related to plant growth promotion. You can see that here we have worked on Mesorhizium deaminase, and I published for the first time in 2017 in computational biology and chemistry. Professor Mondal has already seen some sort of slides that there are some structural and functional analysis of microbial proteins, which can be done just taking the amino acid sequence from the databases. There are some other papers of my bioinformatics that is published in different journals, spin journal, inspired journals. Now, in a short, I am presenting that there are how the heavy metal sensitive bacteria reacts with the heavy metals, and how the heavy metals resistance bacteria reacts with the heavy metals. You can read out from Dave et al. 2021, and I can share the paper also with you, that how the bacteria, the heavy metal resistant bacteria are capable of tolerate such heavy metals. It can be producing such exopolysaccharide. It can be by chelation by metallothionein. It can be some with membrane-bound transporter, which extrudes the heavy metals within the cells if reaches the certain threshold levels. So there are several types of strategies. This is a very complex phenomenon. We can also share this in later also. And you can see there are some operons, ARS, RDABC operon, CZC, RDABC operon. This operon is actually multi-metal resistance operon, which efflux proteins like, efflux, these are efflux proteins which extrudes the cobalt, zinc, cadmium, etc. There are some nickel resistant operons. There are operons for mercury resistance, lead resistance, etc. So what is my conclusion? After doing my more than 7 years of research experience, I am just learned from Professor Maiti and from Professor Mondal and from other some scientists also, that phytobeneficial rhizobacteria or PGPR are natural and valuable resources for sustainable agricultural practices. Unlike other microorganisms, PGPR are unique microflora that has capacity to cope up with both abiotic and biotic stresses. These adaptive strategies are crucial to establish a mutualistic relationship with plants. Omics approaches are worth to incorporate for next generation bioremediation studies and to understand the fundamental knowledge aiming to genetic engineering approaches. So I am happy to share a slide with my which I have attended in NBAIM, National Bureau of Agricultural Important Microorganism. This is Professor Sakshan, you can, of course, you know, and this is Sunil sir, who helped me a lot to whatever bioinformatics I have studied. Dr. Sunil sir have helped me a lot. Publishing my first publication in computational biochemistry. We have some a WhatsApp group also. We are connected with almost all people in a WhatsApp group. We have chats with them till now. And this one is that where I have started my research, what the starting point of my heavy metallist and PGPR knowledges. He gave me, Professor Maiti have gave me a dissertation on chromium resistant PGPR, and then I have started. These are my fellow, my lab mates. Here Sanjeev Da, Pallab Da, in notice is not in the frame, and also in Sayanto, who helped me in some academic studies. And this one I'm with Professor Mondal. I am really honored to have to get in touch with him. He's a nice person. I can say he's a nice person. And these are my lab mates, including Linity, Sohinity, Sucheta, and Sriti. I have also, I am happy to share that I was selected as Foundation Postdoctoral Fellow in 2020. Just the COVID has begun. So I have to leave this, I have to reject this, because due to the COVID only. So I have just proposed that can heavy metal resistant plant growth promoting microorganisms isolated from the rhizosphere or metallophytes be used in the remediation of wastewater? May we use the PGPR as wastewater treatment? So Professor Pablo Cornejo was greatly interested in my project and selected to and accepted to work with me. But I have in my mind with the science. I have mind with me in science. I will definitely work if I get any funding or position in future also. This is University de la Frontera, which I don't know I will go or not any day. Or currently I am working as topic editor along with Professor Pablo Cornejo. Whatever the nice people I meet in life, I am I want to work with them because so I have accepted Professor Pablo Cornejo. I am very pleased to work with him, and also of course my present guide, present mentor, Professor Narayan Chandra Mondal sir. This is a special issue in frontiers in microbiology and frontiers in crop biology and sustainability. The submission is open. So finally, I would like to conclude my slides with special thanks to Dr. Falguni Mukhopadhyay, sir, the chief patron of the webinar, Bidhan Chandra College, Asansol, Dr. Sumen Chakraborty, IQSC coordinator, Department of Botany, Bidhan Chandra College, Dr. Amit Banerjee, coordinator, assistant professor, Bidhan Chandra College, Kasturi Chatterjee, and of course, Kasturi Chatterjee and Anusha Bandhavadhyay, who insisted me to present a lecture in this series. And finally, I would like to thank farmers. Apart from Almighty, the actual soldier of this agricultural field are the farmers. Whatever we are doing in as a scientist, as a researcher, as a professor, but the actual soldier are the farmers for which we can eat, or we can whatever we are for the farmers. Thank you very much.
Thank you, sir, for sharing your research experience with us. And I would also like to convey my gratitude to all our esteemed speaker to making this webinar a success. Now I request Dr. Srinita Ghosh, Faculty of Zoology Department, BC College, to convey the vote of thanks. Over to you, Srinita Di.
Thank you, Kosturi. It's an honor to have been asked to offer a vote of thanks on this national webinar titled Current Progress in Plant Biology, Implications Towards Crop Improvement, organized by the Department of Botany, Bidhan Chandra College, Asansol. On my own behalf, a big thanks to the organizing committee for this. No event can be successful without people who dedicate their resources and time to make sure that everything is faultless. On behalf of the organizing committee, I convey my deepest gratitude and humble thanks to our respected principal sir, Dr. Falguni Mukhopadhyay, for extending his kind cooperation. We look forward to get such guidance as well as active managerial help in the future from him. On behalf of my college and the entire management group here, I want to extend my generous thanks to Professor Narayan Chandra Mondal, Dr. Sanjeev Pandey, Mr. Uday Shankar Ray, and Dr. Krishnendu Pramanik for sharing their graceful words as well as valuable time with us. I'm also confident that we shall be honored by their gracious presence once again and would love to be enlightened in the future by their wisdom. I take this occurrence to thank the IQAC coordinator, Dr. Shoumen Chakraborty, and as well as Bursar of BC College, Dr. Sudip Kumar Bera, for their leading supervision and valuable suggestions to the furthering of this webinar. Thanks to the conveners of this webinar, Ms. Kosturi Chatterjee and Ms. Annesha Bandyopadhyay, for their appreciating involvement in the willingness they have expressed to organize this program. A heartfelt thanks to Dr. Amit Banerjee, coordinator and technical advisor of this webinar, for taking care of all the troubles in arranging this virtual platform for the same. I convey my thanks to the non-teaching staff of the BC College in helping the organizing committee whenever required with their continual support. I would also take the opportunity to thank all the participants attending to this webinar for representing their valuable views through feedback. I, Dr. Srinita Ghosh, hereby declare the closing of the one-day national webinar with the end of this valedictory session. Thank you all once again. Please do take very good care of yourself and stay safe.
Thank you all.
Sir, can you appear in screen? I can take a screenshot.
I'm already appearing in screen. All the esteemed guests are requested to please put on your camera so that Annesha would take a screenshot. Please, sir.